people hear me okay? Okay. So here is some climate change models by Riparian et al. And basically here we have two iconic plants, saguaro and pinyon pine, the most common pine in the southwest. And here you see the current distribution, the projected distribution of saguaros. And you'll see that those distributions are non-overlapping, so how are they going to get there? Here we have the current distribution of pinyon. By the end of the century, the projection is that they will basically be extirpated from Arizona and far more fragmented. Now, they are just models, and so you could make the case, well, I don't believe it. You've got to show it. And that is happening now. Studies are coming out all the time. One of the most recent ones looking at Whitaker and Nearing's 1963 surveys and revisited them 48 years later. And basically things like 70% of the plant species had changed in their distributions on an elevational gradient. Things like alligator juniper had shifted up, had died out in the lowest 1,000 feet of their elevation. And a 1,000-foot shift is equivalent in elevation, is equivalent to a 150-mile northern shift in latitude. So this is happening now, and we really need to be on top of it. And particularly when you're dealing with long-lived organisms like many of us are with trees and shrubs, this isn't something we can put off and just say, well, we'll fix it 50 years from now. We need to be really being proactive in dealing with this now. And so genetics is really can be a major player in this and can provide some real solutions. And we know it works. We have thousands of years of evidence of this in agriculture. We develop, we identify new genetic lines that can really survive in different environments. And we're going to be forced into applying it into wildland systems. And so here's an example of a common garden where we have 4 different genotypes. Each of them have been replicated clonally about 20 times. And you can see genotype number 1 is doing very well. Genotype number 2 is also doing very well, but it's just growing at a much slower rate. Genotype 4 completely died out. Genotype 3 intermediate in its performance. And the good news is there's a tremendous amount of genetic variation that naturally exists on the landscape. And this next slide shows you about 80 studies that have come out of our research group in just the last decade, showing just how much genetic variation there is in traits that are really important to land managers. And so, like with plant growth, 25 to 75% of the variation in growth rate is genetically based. And then when it comes to biodiversity, 39 to 78% of the variation in biodiversity is related to the genotypes of individual plants. And so it doesn't matter whether you're looking at bacteria, insects, spiders, birds, mammals, lichens, endophytes, pathogens, mycorrhizae. They all show these genetic-based relationships. And then water use efficiency, the understory plant community, carbon storage, trophic structure, network analysis, nutrient cycles, soil feedbacks, community stability, even the effects of genetically modified organisms on communities. So the good news is there's a tremendous amount of genetic variation. We can use it in wildlands just like it gets used in agriculture. This will is becoming a powerful tool for land managers. Okay, so with in riparian habitat specifically, we've identified 5 major drivers that affect cottonwoods and willows. Number 1, the use of genetically appropriate stock for future climates. And we can really identify what stock can be used, is best to use under a 3-degree change or a 6-degree change or any level. Number 2, stock that does best in competing with tamarix and tamarix-altered soils. Number 3, the use of appropriate mycorrhizal nutrients. The preparations you get commercially are virtually worthless. They're not keyed in on your particular species, and they just don't work, and they won't survive in nature, so you're wasting your money using them. Plant adjacent to willows that act as nurse plants. Was a nurse for a cottonwood and actually will increase its performance. And then using intact communities that are co-evolved. Right now we mix and match everything, but it's not a good, we can do better. Okay, so I'll go through each of these things. The first one I'll talk about is climate change. And so the National Science Foundation about 4 years ago gave us $4.5 million to set up a common garden array. And this is set up on an elevational gradient in different soil types. And as you go up or down in elevation, there are very predictable changes in climatic conditions. It gets hotter as you go down, it gets drier as you go down. And so by planting the same populations, the same genotypes, the same combinations of species in multiple gardens along the way, you can, by planting them at lower than their current distribution, you can expose them to the climatic conditions of the future. And so we've done that, and we have a lot of backing on this as a real quantitative approach in identifying what's going to do best under future conditions. And so our participants in this are the Forest Service, National Park Service, BLM, BOR, TNC, Game and Fish, Babbitt Ranches, Grand Canyon Trust, and the Arboretum at Flagstaff. And the reason they're supporting this is because we've embedded our field trials in their lands, and so they have the opportunity to learn from our experiments and have adapted, in an evolutionary sense, land management on their adjacent land. Okay, so here's an example. Here's a common garden at the upper end of Fremont's distribution at about 1,500 meters. Here's a common garden at the Cibola National Wildlife Refuge in southern Arizona near sea level. And we have 16 different populations collected throughout the state of Arizona, and the same genotypes growing reciprocally at the upper end of their distribution and at the lower end of their distribution. And this shows the data from this. And so basically, you see productivity as a function of transfer distance. Now, productivity, it could be mortality, any number of metrics that you might want to choose. They are related to one another. And productivity is good because productivity is correlated with biodiversity. And so that's why I show it here. The x-axis is transfer distance, not in kilometers or miles. It's transfer distance in terms of temperature transfer distance. And so a zero transfer distance means in the low garden that you've taken a low elevation population and moved it to a low elevation garden. A transfer distance of 6.5 degrees centigrade means you've taken a high elevation mountain population and moved it down to into the desert where it's 6.5 degrees hotter than its home site. Okay, so what you should see here is that low elevation populations do best at low elevation. High elevation populations do best at high elevation. Okay, so what this means is that they're locally adapted. Okay, so the important thing to remember here is what is locally adapted today will become locally maladapted tomorrow. Much of the stock that we're planting out in restoration trials is basically going to die based upon the climatic projections that exist today. And there's a lot of evidence to really back it up. We really need to be worried about this and take this into our restoration plans because so much of the stock is just simply not going to be adapted to the future climates of tomorrow. Okay, and so there's so much money at stake. We can't, again, we can't afford to screw it up because, like in the lower Colorado River alone, $626 million is being spent. And so we need to plant for tomorrow's conditions, not just for today's conditions. Okay, so this show you where do you go to find the populations for tomorrow. So let's say we're at a high elevation site and the models show that it's going to increase by 3 degrees centigrade. All you do is slide up the scale 3 degrees, and these are the populations where we should be planting today at high elevations, anticipating what it's going to be like tomorrow. And if it increases 6 degrees, then we slide up the scale 6 degrees. If it's going to increase 10 degrees, well, we've probably exceeded the physiological tolerance of the species, and we're going to need to be planting something else that is an ecological equivalent. Okay, this is a very quantitative way of, it's basically cost effectiveness. If you were in agriculture in the Midwest, you wouldn't dream of planting stuff out there that you didn't know how it was going to perform. They have field trials. There's so many millions, billions of dollars at stake. They do the field trials. They figure out exactly what's going to survive, not only for today, but for tomorrow. And so we really need to use this sort of approach to predict what we should be planting now when these long-lived plants start to become reproductively mature and regenerate decades on down the line. And the longer the lived species, the more important it is to take this approach. Okay, number 2, genetic stock that does best in Tamarack-altered soils. Okay, there are these legacy effects of an invasive species, and we just shouldn't be planting out there randomly because different genotypes of plants have different competitive abilities and the ability to tolerate the soils altered by tamarack. Okay, so here is a pristine site of cottonwoods with no tamaracks. Here is a tamarack-infested site with just a few remnant cottonwoods surviving. Unfortunately, this is a pretty common situation. Okay, so this would normally be where the propagation stock comes from for restoration. It's pristine, it's easy to get to, it looks good, so it ought to be good. It would be the logic. But we made the argument that this isn't really the stock we should be using because these are the toughest individuals on the block. They have somehow managed to survive with being surrounded by tamaracks, and that they should be genetically different than these. And when we do the genetic analysis, we find, yep, they really are different. These are genetically different than these. And so to test the hypothesis that ones from tamarack-infested soils would actually do better, we collaborated with Arizona Game and Fish and Arizona Forestry. They bulldozed about 40 acres, and then we put in 10,000 plants, and then both cottonwood and willow, and you can see the tamaracks are coming back really fast. And the next slide shows their performance. And basically what we found is that indeed the ones from these tamarack-infested sites outperformed the ones from the pristine sites, and by about 20%. For both Fremont cottonwood and coyote willow, they both had about 20% increase in survivorship when they came from these sites, and they had about a 20% increase, 20 to 30% increase in growth rate when they came from these sites. Okay, so just using a genetics approach, we can do better. And then we need to inoculate with appropriate mycorrhizal mutualists. Here's a slide from a study from our group of Meinhardt and Gearing. And basically what happens is that tamaracks inhibits the mycorrhizae of cottonwoods and willows. And this shows that in the presence of tamaracks, the beneficial arbuscular mycorrhizae and the ectomycorrhizae decline by 50 to 66%. And basically in tamaracks thickets, there will be no beneficial mycorrhizae left for cottonwoods and willows. And these are really important mutualists. Okay, the fourth thing is that we need to plant adjacent to nurse plants that can benefit the cottonwoods. And so here's a field trial where we have willow planted adjacent to cottonwoods, and then we compared how well the cottonwoods did when they were adjacent to willow versus another cottonwood or no other plant at all. And we found cottonwoods paired with willows have greater biomass than those growing alone. And we found that male willows are better nurses than female willows. So males are more nurturing in this case. Okay, and then lastly, we need to use intact communities in restoration. Um, and so here's a field trial in which we collected cottonwoods and willows from the same site, and we grew them together in a restoration trial. And then we called those sympatric plantings. And then we had allopatric plantings, which were mixed and matched from different sites, cottonwoods and willows. And we got in these replicate studies, overall shown on the right, um, very significant differences. The sympatric plantings outperform the allopatric plantings, and it's largely because they're co-evolved. They become genetically adapted to one another over time, and when you mix and match, then you break up all those co-evolved relationships. Okay, and this also extends to the arthropod community. When they're sympatric, we have greater arthropod richness and abundance than we do with the allopatric plantings. And this is really important. This study was done relative to the southwestern willow flycatcher. Okay, so what we're trying to do is merge all of these 5 factors together in simultaneous. We take everything we know about genetics and nurse plants and these mycorrhizal associations, and we combine them in field trials and compare them with the current best strategies or traditional strategy of using only local stock. And basically, we're now using them all together. And in this field trial, funded by the Nina Mason Pulliam Charitable Trust, Babbitt Ranches, and NAU, and we think we do a lot better. We're very much hoping to do this in other places in the West to see how we can benefit from these approaches. And one trial doesn't tell the whole story. We need to do this over and over again, and if there are any in this group that are interested in a collaboration of this kind, we'd be very interested in doing so. I'd like to end by just simply acknowledging our collaborators, our researchers. Their skills range from genes to ecosystems and microbes to vertebrates. We've been well-funded from the National Science Foundation. We have a lot of land managing agencies involved, and we think we're really making some very good progress on a genetics-based approach for dealing with a very important environmental problem. Thank you very much.
Thank you for your talk. I'm a big supporter of your message. I was just wondering, in general, what do you think about but at least taking into account genotypes that are not only locally adapted to predicted climates, but ones that are more plastic and able to handle unpredictable change?
Absolutely. Actually, there's a very strong genetic component to plasticity itself. It's often not thought of that way, but there is. And so some plants are almost invariable. They are not plastic at all, and then there are other genotypes that are extremely plastic, and that's a heritable trait. And so if it's heritable, we can work with it just like agronomists deal with it in agricultural systems. So absolutely.
First of all, thank you for such an enlightening and well thought out talk. It's absolutely brilliant. Secondly, your genetics work, or is it simply common gardens, or are you also doing sequencing studies? It looks like you had some molecular geneticists.
We do. And so we're identifying the genes associated with biodiversity, the genes associated with water use efficiency. The genetic cottonwood was the first tree to have its genome sequenced. We now have a 1,000 genome project where we can identify the genes associated with it. It's amazing what the future is going to bring in terms of identifying the genes that are related to so many things that are relevant to not only the survival of the species, but traits like biodiversity that land managers really should select for, I would argue. So yes, we're including that.
Oh, when you were talking about degrees Celsius hotter, and you said, for instance, this is 6 degrees hotter, are you referring to annual high, annual low, or just an average temperature in the area?
Yeah, that's a really important distinction. Um, Heat wave effects are being shown to be increasingly important and maybe more important than the actual means. And so when you get a heat wave come through, these temperatures, right now we're already at heat wave increases of about 3 to 4 degrees centigrade in extreme events. And in cities, it's amazing, the extremes in the cities are as high as 27 degrees Fahrenheit hotter in cities than they are in the adjacent rural areas. And so, we need to be breeding for a whole different group of genotypes of organisms that can survive the city environment, because we've already exceeded most of the climate change projections by a large amount when we start talking about cities. So we need, yes, we need to separate between means and extreme events, and between cities and rural areas, and there'll be clearly a genetic component to all that.
More of a curiosity, does your website from NAU or the SIGA website, a place where we go find more of the papers that you were referencing, and
yes,
to go dig into it a little more?
Yeah, SIGA is relatively new, and the website is growing as we get more users. And SIGA is set up for anybody to study anything from microbes to grasses to to trees, and so we definitely want to encourage its broader usage. And the power of any common garden approach is the larger the array, the greater the resolution. And so that's why we need collaborations in other states and countries. There's a movement underway to replicate this whole approach in Australia and France and Norway, areas around the world, because this is a global problem, not just a Southwest problem. And so by having more of these sites and more of these collaborations, if we just set them up similarly in experimental design, then we can really compare across states and across continents really compare our data and show exactly, with much greater accuracy, what's going on.
I've got a question about the mycorrhiza mutualists, the ones that are pertaining to the willows and the cottonwoods. Is there any studies that have been done regarding how long it takes for those things to reestablish themselves? If you were to come in and plant poles immediately after tamarisk removal, is there a period of time where they simply will not live because the mycorrhiza can't grow?
We have anything that comes out of a greenhouse is going to have a greenhouse mycorrhiza, and that mycorrhiza will not survive in the wild. And so this is one reason why we should doubt a lot of greenhouse studies. But by inoculating with soil from the sites, when you plant that plant, is the ideal time to do an inoculation in a restoration program.
Okay, thanks.
Right at plant. Yes, I'll be here today and tomorrow.